Hello, welcome back. Our next speaker, Elaine Fuchs, has been key to our understanding of our largest organ, which is the skin. Professor Elaine Fuchs is the Rebecca C. Lansfield Professor at the Rockefeller University and a Howard Hughes Medical Institute investigator. After studying at the University of Illinois and Princeton, she became a pioneer in skin biology and stem cell research. Her studies of how stem cells regenerate tissues and respond to stress have provided fundamental insights into wound healing, cancer, and aging. She has received many science highest honors, including the L'Oreal UNESCO Award, the Gardner Award, and the National Medal of Science. Her talk is titled Tissue Stem Cells Coping with Stress and Learning from the Experience. Please welcome Dr. Fuchs.
Okay. Can everybody hear me in the back too? Okay, great. All right. Well, let me just begin by saying that it's great to be back here. Fernando invited me here. We were trying to figure out, it was probably about 15 years ago, for an Ochoa lecture. And it's always good to be back. I feel like the whole building is different. Everything's been different, but the science has been just exciting all the last several days. So really, you guys are on a good track. So I am a skin biologist, and it's always nice to try to start with one of those things. I've heard these before. Scientists, we learn quickly that there's problems. Okay. So let me start by really emphasizing that there's no other organ of our body that has this kind of diversity throughout the course of evolution, much of which has been evolving to be able to deal with extremes of environment, whether it's a porpoise in the water or basically a kangaroo out there in the desert or whatever it is. And so there's an enormous sea of interesting questions that we can ask about the skin, and just the fascination about how our skin has evolved to be able to deal with these different extremes in temperature. And so now let's consider our current climate and what happens when our climate is now changing faster than it ever has been before, where we're dealing with plants that are altering the pollens and allergens that they're generating. We're dealing with carbon emissions and air pollutions that are becoming more complex and numerous, where the ozone layer is thinning. We are dealing with existing pathogens and allergens that are shifting their territories, as we heard earlier this morning about dengue virus showing up in Texas, where it never did before. We're learning about new pathogens, allergens, microbes, and pollutants that are emerging as we're painfully aware by the recent pandemic. And so when you think about it, bearing the brunt of these environmental changes is the epithelium, and particularly the epithelium of the skin. It's our body's barrier to the outside world. And when we consider the variety of different stresses that we are giving our skin on virtually a daily basis, it's not only ultraviolet light and mechanical stress, but wounds, noxious agents, commensals, as well as pathogens, and then temperatures and pH extremes. You go outside on a warm day and you come back in, and your liver is still the same temperature, but your skin surface is something quite different. And so the whole course of epithelial development and differentiation has to be able to be compatible with this. The stem cells that are responsible for making and repairing the body's surface have to be able to be poised to be able to rapidly adjust to these various different stresses, and to do so over a lifetime. And so what we know from the course of our own studies and those of others are that if epithelial stem cells malfunction, either to make the proper barrier or in communicating with the immune system upon a barrier breach, then chronic inflammation occurs. And what we know is that just a couple of years ago, the World Health Organization ranked chronic inflammatory disease as the greatest threat to human health. There are many different risk factors. I've named some of them already: mechanical stress, pollutions, allergens, but chemical irritants, aging, obesity, smoking, Western diets. The list goes on and on. And in addition, we know from the work back in the 1860s that there are links between inflammation and cancer that have been postulated and are becoming an increased reality. And so, what are we talking about dealing with chronic inflammatory disorders? These are common disorders in the skin. They include psoriasis or atopic dermatitis, chronic wound healing, but also bladder infections, asthma, inflammatory bowel disease. And they all have in common certain features. One is that they're triggered by some kind of irritant. It could be a chemical infection, mechanical trauma. Infection or inflammation often comes and goes in these diseases. And what we know is that it often recurs again, sometimes years later, in the same spot as before. And I think many of you in the audience have experienced this, and yet it's really telling us that our tissues have some kind of memory that they've experienced inflammation at some earlier time, and it could be years ago. Another interesting aspect is that recurrence is often accompanied by increased severity. And finally, and keep this in mind, the secondary bout of inflammation can be triggered by a different irritant than the first one. And I'll get back to each of these as we go along. And so back about a decade ago, Samantha Larson, who is a graduate student in my laboratory, and Shruti Nayak, who is a postdoctoral fellow in my lab, wanted to know what was causing the skin to remember a previous inflammatory experience. And so they did a very simple experiment. They just took a laboratory mouse and they treated it with one side of its back with a drug or topical stimulant called imiquimod that elicited a type 17 immune cell response through the dendritic cells, which trafficked to the lymph nodes, which basically produced cells that were spewing out IL-17, and it's the epithelial epidermal stem cells that have the receptor for IL-17. And so that basically caused hyperproliferation within 6 days in the skin, and a cross-section just shows you tremendous amount of hyperproliferation relative to the control side of the skin. So in this particular stimulus, there's no systemic or major systemic component that is involved. So now they waited 30 days later. The pathology had returned back to normal, but now if they wounded the skin on each side of the animal's back, they found that the side that had been exposed to inflammatory stimulus a month earlier, was now healing its wound faster than the other side. And so this was giving us the sort of memory that we were looking at, and we wanted to know more. We did this experiment on the background of all sorts of different types of stimuli. We used Th2 stimuli, we used tumor promoting agent, we used wounds, we used a yeast infection. And in all of these cases, when you let the pathology resolve and then introduce wounds, you find that the side that had experienced the inflammation always healed faster. And so what this is telling you is that it's probably not an adaptive immune response of the sorts that you've heard about earlier at this meeting and earlier this morning. And in fact, we could repeat this experiment on a RAG2 null mouse that's missing all B and T cells. And again, the same experiment, getting the inflammation, letting it resolve, wound, and the wound still healed faster. So this is not adaptive immunity per se that's causing this response. We thought maybe it was the macrophages because they had been implicated in something called trained immunity, which was an epigenetic response. We also showed that the macrophages were dispensable. We then actually knocked out the cells that were producing the IL-17 that was stimulating the inflammatory response in the first place. If you knock out those cells initially, you can't get the inflammatory response. But if you first allowed the inflammatory response, allowed it to resolve, and then introduced a wound, you found that still, in the absence of these cells, the wound still healed faster. So again, this is not coming out to be immune-centric. And it was only at about that time where we were convinced that it was immune-centric and it wasn't, that we began to realize we work on epidermal stem cells, and maybe these cells are the cells that are harboring this memory of chronic inflammatory, an inflammatory experience. And that turned out to be the case. And so in the epidermal stem cells, the epidermis is nearly normal after the inflammation resolves. The skin looks normal. As I showed you, the pathology looks normal. As I showed you, skin proteins are normal. And even the skin messenger RNAs are normal. And right away, that is telling you that it's suggestive that the response or the memory is intrinsic. Because had it been something like scarring or some immune cell that is hanging around or the microbiome that is changing, you would have expected that the stem cells would change their program of gene expression if something was hanging around for the long haul. And yet we're seeing the messenger RNAs are returning back to normal or largely normal. And so keep that in mind too, because our studies now have suggested and indicated that this is an intrinsic response that's happening in the epidermal stem cells that is triggered by the microenvironment to begin with. Tucked inside the nucleus, we found hundreds, in fact, thousands of memories of the inflammatory experience. And these were epigenetic changes in the chromatin that weren't there before the inflammation, but were lingering up to 6 months afterwards, which is about 5 to 6 equivalent human years. And so now we're also dealing with the time frame of why years later that inflammation about an inflammatory experience can happen again in the same spot as before. And with the epidermal stem cells, in contrast to the immune cells, these are stationary, resident within their frame of the tissue. So again, why the disorder would come back again in the same spot as before. So are these really functional? The way in which we described or determined that was simply to take out these peaks. The red peaks are the inflammatory experience. The gray is the control. We cut out some of these peaks that lingered up to 6 months later after the inflammatory experience, and we used them as enhancers to drive EGFP. We made 3 different transgenic mice in our rapid transgenic methodology of simply packaging the gene into a lentivirus and then simply bathing the living embryo with lentivirus, and at a stage when the skin epithelium just exists as one single layer. So it's essentially like taking a petri dish and adding lentivirus to it to transform the cell, but instead this is a living embryo that we can then allow to grow up into an adult animal and do experiments. And so when we do that, what we find is that no inflammation, no activity, you now apply imiquimod, and in all 3 of these cases, we see activity. So these are inflammation sensors. They're sitting in the mouse's chromatin and probably in your chromatin after an acute inflammatory response. And that is happening at an open state for long after the inflammation has subsided. We also looked at what about the genes that are associated with these inflammatory peaks and found that those genes tend to be activated faster on the next experience, in that case, a wound. And so we now wanted to know, how does this work? And so I'll just summarize for you data that we had published several years ago, where it's now work of Chris Cowley and Rod Sajoth in my laboratory, along with Samantha Larson. And in this case, what we did was simply to start with a closed chromatid state, essentially in the naive state, no inflammation. And these genes that are associated with these memory domains are in a closed chromatin state. So we need to activate the chromatin, and the way in which this is done is with the activation of phospho-STAT3, which is downstream of the IL-17 response. And what happens then is phospho-STAT3 can bind to nucleosome-bound DNA, and it opens up the chromatin. That's not enough to establish the memory. We showed that we needed another factor, and that second factor turns out to be FOS, which is also activated upon inflammation, upon many different kinds of inflammation. And FOS then heterodimerizes with JUN, which is endogenous, to form an AP1 transcription factor, which then remodels the chromatin, causes or recruits RNA polymerase and allows transcription of the associated gene. I'll also mention that there are multiple histone modifications that we identified that happens while the chromatin is open. And while the chromatin is open, there are now additional sites for stem cell transcription factors that couldn't see the closed chromatin before. Now they can, they come in and bind, and they now are also part of this open chromatin complex. So now what happens after the inflammation is subsided? We no longer have STAT3, we no longer have FOS, but what we do have are the stem cell transcription factors and some of the histone modifications, and the transcription is now in an off state. So we did a lot of experiments at the bench and a lot of cut and run and ATAC and chromatin profiling that basically, and genetics that basically summarize what I've summarized here. And so now think about this. All you need now to reactivate these genes is simply any kind of stress that will induce FOS, which is mostly any kind of stress, because the chromatin is already in an open state. So you no longer need STAT3 for this secondary memory recall. And so now all we need to do is activate with any kind of general stress, activate FOS, brings in JUN, remodels the chromatin, and recruits again RNA polymerase. So you start to now see how activating transcription is faster. It's more robust in memory recall because the chromatin is already in an open state. And so this is essentially summarizing what I've just stated to you in terms of understanding how is memory established? How do we get these memories of an acute inflammatory experience? And it also is suggesting how those memories are kept in an open, poised state for the next assault, and how upon the next assault, there is a more robust response because the chromatin is already open, and how it can be general types of stress that will induce that secondary response. So that's why we can do a wound, for instance, instead of a Th17 inflammatory response for memory recall. And so at this stage, we had a pretty good understanding of at least the front and the back ends of what is memory. And at this point, we really wanted to know, is this just something special for the skin epithelial stem cells, or how broadly is this mechanism that I've just described to you? Well, we now know from that it's not just the skin stem cells, but it's also the airway epithelial cells, it's the intestinal stem cells, and it's the blood-producing stem and progenitor cells, all of which have been since demonstrated to have this kind of epigenetic memory that I've described of inflammation. And in fact, when we took our data of paradigm of how this works, it might not necessarily be STAT3. A different type of inflammation may trigger STAT1, or maybe it triggers NF-kappa B. But when we looked at a variety of different databases, including CD8 cells, which you heard about certain types of CD8 cells this morning, of different types of viral-induced cells, of natural killer cells, of dendritic cells, we look at the data and analyze it in the confines of the model that I've just described to you. At the front end and the back end of all of these different systems are AP1. And so FOS triggered by FOS and JUN. And so these data suggest that the mechanisms that I've described to you are actually general mechanisms that our cells have. Curiously, when you think about the way in which our neurons store and recall memories in the brain, Eric Kandel predicted a long time ago that AP1 transcription factors may be important in learning and memory. And I don't want to push it to my neurobiologist friends. They always don't like that comment. But I actually think it's worthwhile thinking about, because it really raises this general question of whether all of the tissues of our body might have the ability to learn from our experiences. And so we next wanted to address a question that we're currently working on. And this question really deals with, do memories erode with age? Remember that an important aspect of the maladaptive aspects of epigenetic memory, those aspects that deal with chronic inflammation or potentially cancer, that those aspects are there that might be realized many years later. And so it's really this mechanism of memory longevity that is really critical with understanding what is the pathology associated with epigenetic memory. And so this is the question that we've been spending much of our time lately. And we've been joined by Luis Soto Ugalde, who is a joint graduate student between my laboratory and Donna Pear's laboratory, who's an expert computational biologist. And we've been using machine learning to help guide us through the process of really understanding the details of epigenetic memory. We've followed this out now, and this has taken us quite a few years to be able to do these experiments, as you might imagine, to see just how long memory lasts. And we followed it out looking at sampling the skin after an acute inflammatory response up to the entire animal's lifetime. As we're doing these experiments, it's critical to always use an age match control mouse because there are age-related changes, as we and others have demonstrated. But these are changes that are happening from acute inflammation. And surprisingly, despite all of the age-related changes that are taking place, the acute inflammatory changes that are happening appear to be largely independent of the age-related changes, which I think is quite interesting to think about. It's not to say that there aren't going to be age-related changes that impact our ability to develop memories and our ability to store and recall memories, and those are additional interesting questions to ask. But what we have demonstrated is that it's not all memories that last forever. It's only some memories that last forever. And so in following this out, what we found is that what's interesting is that an animal that is 1 year old that had an acute inflammatory experience when it was a few weeks old, essentially has an ability, a beneficial advantage by having epigenetic memory so that it can heal its wounds faster. We know that our wounds heal more slowly as we age. Here's an example where if we've had an acute inflammatory experience, it actually acts as an advantage with regards to wound repair. Another interesting aspect is that while some memories last forever, other memories start to wane over time. And nothing that I explained to you thus far about our knowledge of epigenetic memory can account for this type of phenomenon. And so we've now been using machine learning to ask for our computational biology data to be able to be placed with regards to predictive analysis. And as you heard yesterday, it's only good as a predictor, and you have to have a lot of data and a lot of good data. But then you start to get out predictions that you can then go back and test experimentally. And thus far, I'm a strong believer of AI because the predictions that we've come up with are turning out to be remarkably accurate. And so stay tuned to the story. But the crux of this is really, are we going to be able to understand memory enough so that we can start to keep those beneficial memories, things like broader pathogen resistance or aspects of increased wound healing? Those are good memories, but to get rid of the bad memories that are contributing to chronic disease and cancer. And so that's where we're headed on the big picture. We've also looked at just because the skin has this ability to retain memories of acute inflammation, maybe it's got the ability to retain all sorts of different types of memories. And that turns out to be the case as well. In this case, we used wounding as a model, and we took advantage of the fact that if you wound the epidermis, which every time you see blood, you're in the dermis. So you've completely removed your epidermis. If you wound, it's your hair follicle stem cells that are basically being mobilized to move upward and repair the epidermis. So those hair follicle stem cells have to exit the niche for the hair follicle, and they have to migrate upward They enter a wound bed. There's inflammation there, so they obtain the inflammatory memory. But now thereafter, they have to act like their epidermal stem cells, no longer hair follicle stem cells. And what we found is that they're acquiring and retaining memories all along the way. So they have the quicker response to inflammatory stimulus. They have an increased ability to migrate and heal subsequent wounds. If you take one of those stem cells that's now in the epidermis that did this thing and you put it into culture, it can migrate faster than a normal stem cell. And so it's, again, intrinsic effects of memory. There's also increased stem cell plasticity. The stem cells remember that they used to be a hair follicle, and even though they're now just performing as an epidermis. And so that type of plasticity and the ability to migrate and heal faster and the ability to respond faster to inflammatory experiences are all features of cancer and the stem cells that are fueling the cancer. And so we're really now focused on whether these prior assaults to the skin are predisposing us to non-melanoma skin cancers with age. And so I'll just finish then by dealing with additional stress. I mean, as we are existing in our environment, we're not just having one kind of stress, we're having lots of stress, and the stress always seems to be getting stronger and higher each time. So what happens when we start to get excessive stress? And so we started to look at that. And what we found is that there is a pathway called the integrated stress response that activates in these stem cells in response to stress. And each one of the stressful experiences triggers the integrated stress response. And so it's cumulative. And so you can keep adding stress to the epidermal stem cells. What happens is that cellular fate is coupled to the integrated stress response. And that's relatively new finding that we made and published earlier this year. And why would you do things like this? Why would you want to link cell-fate decisions with stress? And the explanation that we feel is likely to be the correct one is that when a tissue has a lot of stress, what it wants to do is to be able to shut down those pathways that are dispensable. And in this case, I'm sorry to say it's the hair follicle that is dispensable, making hair, forget it. But what the cells have to do is to be able to repair wounds. And so that's exactly what happens. You get increasing levels of stress, you're shutting down further and further the ability to make hair. Those of you who are having trouble making hair might recognize that. You should really try to limit the amount of stress that your life is experiencing, because essentially that's the bottom line to the story that I wanted to mention to you. I think it's a really these are quite, I think, interesting aspects of biology. So just to summarize then, all tissues of our body have to cope with stress. They have to repair wounds. Since the 1930s, it was known that infants that are vaccinated against one bacterium develop protection against another. It was known that plants that survive one pathogen are often resistant to other pathogens. And what epigenetic memories do is they afford a means to apply the lessons learned from stressful encounters to those future encounters that our tissues are faced with. They can have both beneficial as well as deleterious consequences. And the challenge moving forward really is, how can we keep the good memories and get rid of the bad memories? And ultimately, it is a problem of survival of the fittest. So those are not the people that worked with me. These are the people that worked with me. And there's one Spaniard in the fray, and that's Jorge Almagro, who I think originated from Madrid. So I'll just leave it at that and say thank you again.
Questions?
Very nice. I wonder if, because you talk about good memories and bad memories, if it could be good memories for one biological process, but at the same time, bad memory for other. I mean, you said that you are doing this now, but in the case of inflammatory response and wound healing, that you have rapid wound healing, what about cancer, DMBA, TPA treatments? I mean, could be worse, no? In this case.
Yeah, we're doing those experiments, and I think it's an interesting question because there's long been this link between inflammation and cancer, but it never has really fit well because the whole notion of inflammation you would think is counter to malignancy. But the whole notion of epigenetic memory of inflammation is actually likely to be beneficial with regards to cancer, maladaptive with regards to epigenetic memory. And so we're doing exactly those types of experiments of saying, if we treat with an inflammatory stimulus, say TPA, to begin with, allow memory to be established, and then treat with a malignant oncogene, does that progress to cancer? If so, it's epigenetic memory, or can we actually have the inflammation after the activation of the oncogene? And my guess is, my prediction would strongly be that it's epigenetic memory that's the driving force of cancer. And the other way one can look at that is the genes that are being activated, and are those actually oncogenes? And many of the wound response genes are.
Thank you.